So let's study about the staining. Staining technique is used to stain the bacteria, microorganisms. Basically, we used it for the bacteria only, right? So gram staining and gram staining, acid fast staining are the two main staining from where the question arises and comes in gpat and NIPER. So let's see. Gram staining is a method in which we identify gram positive and gram negative bacteria, gram negative and gram positive. So what is the principle of it? So it gram stain the cell wall, right? It will stain the cell wall of the bacteria, and which is composed of peptidoglycan, right? Peptidoglycan. All right. After that, what if the actual if the peptidoglycan layer is if it is thick, then it is gram positive and there is no extra layer of lipid which is present in gram negative bacteria, and in gram negative bacteria peptidoglycan layer is thin. Hope you got it now let's study the procedure of the gram staining. So the primary dye we use here is a crystal violet. Crystal violet is the dye, or methyl blue is the dye which we use in gram staining as a primary dye. It gives the purple or blue color, right? Then we use a moderant to fix this dye inside the bacteria. So we use here moderant, that is iodine, and it works as a entrap the color. So it will entrap the color inside the bacteria. And then we use decolorizer. So we use alcohol and acetone that will make pore टू पास द डाई आउट, ठीक है, जो हमारी डाई को बाहर निकल में हेल्प करेगा. बट नाउ इट इज़ डिपेंड ऑन द बैक्टीरिया, विच टाइप ऑफ बैक्टीरिया इट इज़, एंड इट विल इट लेट और नॉट टू डाई कम आउट, ठीक है. सो ग्राम पॉजिटिव बैक्टीरिया में क्या हुआ? पॉजिटिव मीन्स देयर आर थिक लेयर, राइट? थिक लेयर इज प्रेजेंट ऑफ peptidoglycan and there is no third layer of lipids, right? So for that, in gram positive bacteria there is thick layer of peptidoglycan, so acetone and alcohol is unable to pour through it and the dye remain inside it, and it will cause a purple or blue color which is the primary color of the primary dye, right? That means it is gram positive bacteria if it do not release these dye out. Okay. Then is gram negative bacteria which has a thin layer, right? And acetone and alcohol is able to pour through it and dye will come out. Then we use second stain which is a counter stain or our secondary stain, right? Safranin. Basically we use here safranin. Carbol 15 in some time, but mainly is safranin, and which is in pink or red in color, right? So the gram negative bacteria will contain a pink or a red color. That's why we can differentiate in microscope with these two different of colors, right, between the gram negative and gram positive bacteria. Hope you got it. Next is acid fast staining. Let's do this one also. Acid fast staining. Acid fast staining is used for the cell wall of the which contain mycolic acid. Basically it determine that the bacteria is of mycobacterium class or not. It tells that, right? Mycolic acid is present in mycobacterium like tuberculosis mycobacterium. Okay, then let's see. Acid fast staining is used for this purpose only. Now let's see. So the primary dye we use here is carbol 15. Carbol 15. What? Carbol 15 is the primary dye which is red in color, right? So this acid fast staining is also known as a Jahil-Nielsen method, the scientist name. Okay. Now we use here moderant, heat or phenol, right, to entrap or to fix that color, right, to fix the color of dye in that bacteria. Now we use here decolorizers that to test that it will come out or not, right? So we use here a specific type, like we use alcohol as same we are using in this as well, like alcohol. Now we are using here alcohol plus some amount of acid, so that's why we are saying acid alcohol we are using here. We can use 3% HCl or H2SO4 and plus ethanol, which is alcohol, right? So by the combination and dilute alcohol, or so we are using this solution to decolorize the bacteria with the primary dye, but it depends on the bacteria, right? And it tells about the bacteria itself. Is it acid fast or non acid fast? So acid fast bacteria will retain the color and resist decolorize, and it will be red in color. Hope you got it now. Acid fast, right? Which means it contain mycolic acid. And in non-acid fast, it get poured and lose the color and decolorized. ठीक है, तो वो decolorize हो जाएगा. Then we are going to use here secondary stain or counter stain, right? So in secondary stain we using this. Secondary stain में हम use कर रहे हैं मिथाइल ब्लू और मालचिट green. Basically we use here methylene blue, right? So which gives blue color. Hope you got it. Which gives blue color and differentiate between acid fast and acid non-acid fast. So acid fast will be in red color because it contain which carbylfuchsin, which is red in color, and it release the carbylfuchsin and it retain the secondary stain color, which is methylene blue. Hope you got it. And acid fast staining is also known as Jahil-Nielsen method, which is based on the scientist name. This is how it is used. It differentiates whether the bacteria cell wall is made up of mycolic acid or not, right? Hope you got it. Now let's see this one also, which is based on the visualization of parts of the cell or bacterial cell. ठीक है? So flagella, cell wall, spore and capsule. See, so we are using this. So first is flagella. So this is the flagella and it is negative, means it is acidic in nature, right? So we are using to this we are using here, as it is negative, so we are using here positive stain, right? Positive stain. Like it is acidic, then we're gonna use here basic stain. So I'm just telling you all names we can use here. So basic dye is fuchsin dye, right, which has a positive charge dye and it will attract to the negative part of it and will stain this. Hope you got it. Now we are using spore staining for the spores. Like these are the spores inside the in the membrane of the, or in the cell wall of or capsule of the bacteria. All right, let's see. So first we are using here primary stain. Primary stain is malchite green, so it will cover all, it will be applied on the whole bacteria, but it will remain in only pores. Like it will remain here, 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 and it wash off from here, right, in pores, right, and it will give a green color. Then we want to use secondary stain, right, and secondary stain will be on the cell wall, right, and it will give a pink or red color. So when we see it in microscope, right, we will find that pores will be in green color and whole surface will be pink or red color. This is how we use it for spores. Then we are going to use it capsule. Capsule is like made up, this membrane is negative, right? So here we color the background area of this. So it is negative. We can use positive but not, just to differentiate. This is the capsule we are using. It is the inside cell wall and the capsule is this, and so capsule is also negative. So we will use here another negative stain, right, which will, right, repulsion will be there and no staining will be there. We will color this area of it like of cytoplasm, right? And will and then when we cover the background, so we will find this cell wall like this. Like it is, we are using here cytoplasma is also colored, and some negative stain is also used here, and which through which it has a repulsion. It is not depleting here, so making some space. This is how we can stain the capsule as well. And cell wall is used. Cell wall also is a negative in the nature, means it is acidic, and we use here methylene blue, which is a basic dye. Hope you got it now, all things.